I'm Julianne, this is Hyung-Do, and this is Francesca. We did DNA replication and structure.
Okay, so this is our model of DNA structure, and this is our model of how DNA replicates. So just to explain what everything is, we have yellow and green micanites, yellow for cytosine, green for guanine, and then you can see in both models brown and red Twizzlers. Brown is for the adenine, red is for the thymine. And then we made homemade Rice Krispie treats, and they're the phosphate, and then these little orange and yellow rings you see are the deoxyribose. Our hydrogen bonds are toothpicks. Julianne will later demonstrate with the frosting for our DNA polymerase, and helicase is our cute little scissor because it cuts the DNA and connects with each structure.
So with the structure of DNA, the cytosine and guanine, because they are complementary bases, they are connected with one strand to the other, and then next is adenine and thymine. And this continues, and always the complementary bases have to attach together, and this strand goes in a helix, and then it forms a double helix together.
And we have 3 hydrogen bonds for the cytosine and the guanine, and we have 2 hydrogen bonds for the adenine and the thymine. In addition, we have our okay, never mind. Let's go on to DNA replication. So this is our model of DNA replication. Let me first explain it to you because it might be a little unclear. This here is one strand. This here. Then this here is our second strand. And as you can see, this is coming apart because we have our helicase. It's literally cutting the DNA in half.
And hence our cute little scissors.
Our little scissors. And then we have our, this is our DNA polymerase. And it's frosting, and frosting is the culinary glue. So it's going to add the nitrogenous bases to the original nitrogenous bases. So, yes. So this is here is our replication fork. It's where the helicase comes. It's where the helicase and the polymerase meets. And so we have one strand that is replicating from the end to the replication fork, and then we have one strand replicating from the replication fork to the end. And so this strand that's going from the replication fork to the end, we have something called a primer here. The primer is here to tell the DNA polymerase where to go. It's where to replicate. So it's saying to the DNA polymerase to come here and start replicating here. Hence we have our nitrogenous bases starting here and going this way.
On the other side.
And on this side is starting here and going this way. And then our DNA polymerase, yummy, will come right here and right here.
And it's not going to come off the spoon, is it?
Can. Woo! Yay!
Bomb.